Okay, evening all. This is the next video on mutations, alleles and genetic diversity. And if you're reading along at home, this is on page 15 in the notes, begins with genetic diversity. So in this I will look at how alleles are produced by mutation and link that to genetic diversity. Look at the different types of mutation. Now I'm not going to go into too much detail about the different types of mutation because there are animations available on YouTube that can do it much better than I can here. I'll just point you in the direction of those. But we will have a look at the impact of different types of mutations on proteins, cells and tissues, etc. Okay, so genetic diversity means the many genes that exist in their different forms. So each of your chromosomes is made up of DNA, and along those chromosomes there are genes at different loci, or an individual locus, and they can exist in different forms, and we call those different forms alleles. Okay, so these are alleles. They are the alternative forms of a gene. And those alternative forms of genes, those alleles, are created, originate from mutations. Now we know that a gene is essentially a sequence of nuclear. Let's rub that out. Start again. There's a sequence of nucleotide bases which code for the sequence of amino acids in a polypeptide. And if you weren't sure about that, look back through the transcription and translation videos that I posted last night. Those mutations will change the sequence of nucleotide bases. And if you've got a changed sequence of nucleotide bases, you've now got an alternative or alternate form of the gene, or what is referred to as an allele. We'll go into that in a bit more detail. So here we see two chromosomes. Now this is a homologous pair. Now Mrs. Andrews did a really good video on counting chromosomes, which is a really confusing subject for a lot of A-level biology students, and it might seem simple, but it's actually quite a tricky thing to do. So I'll put a link in the description to Mrs. Andrews' video on this, and you can watch that. But the only thing that I would add to Mrs. Andrews' video, which I think is excellent, is my method for counting chromosomes, and that is to count these things. So in the chromosome there is a centromere, and a centromere is a complex of proteins. Oopsie daisy, where's my other one gone? It's a complex of proteins that, here we go, that form and help join sister chromatids together after DNA replication. So you may be familiar with seeing, don't use that, a chromosome in this shape, in that X shaped. And in one of my previous videos I described this as this is still a chromosome, but it's comprised of a pair of sister chromatids. And those sister chromatids, they are identical. They're made up of DNA, and the strand of DNA on this sister chromatid is identical to that DNA on that chromatid. Okay. In the middle, holding those two sister chromatids together, is the centromere. So if you want to count the number of chromosomes, my advice would be to count the centromere. Now they might not always be present. Okay, so you've got this diagram in your notes. It shows a diploid cell. So diploid meaning that it shows the pairs of homologous chromosomes. So one maternal chromosome and one paternal chromosome. So remember, homologous chromosomes, or a pair of homologous chromosomes, are two chromosomes with the same genes at the same loci. Okay, so two chromosomes with the same genes. That was enough in an exam question a couple of years ago. Some exam mark schemes require this last bit at the same loci as well. Now, the centromeres aren't actually shown on here. So what I sometimes do, even now, just to remind myself, is draw those centromeres on. Because then, after DNA replication, we can see those centromeres are now connecting the two identical sister chromatids together. So if we want to count the chromosomes, well, we have two chromosomes here, so 2n. Want to count the chromosomes here, two chromosomes, two centromeres, so two chromosomes, so also 2n. Okay, right, have a look at Mrs. Andrews' video on that. She explains it much better than I do. Let's go back to the PowerPoint. Okay, so what is an allele then? We've mentioned an allele already as the alternative version of a gene. And they exist at loci. So here we have the gene. Now the gene is the same, it is the allele which is different. So this allele is a sequence of nucleotide bases which codes for white flowers. This allele is a sequence of nucleotide bases which code for purple flowers. Just to reiterate, because this comes up an awful lot and you'll be surprised how many people get this wrong, a homologous pair of chromosomes are two chromosomes with the same genes at the same loci. Got a marking point there. Okay, so let's demonstrate that in a different way. Here we can see a sequence of nucleotide bases, allele number 1, and a sequence of nucleotide bases on allele number 2. We can see here this adenine present on allele number 1 is replaced by thymine on allele number 2. Now how does that happen? Well, the change in that sequence of nucleotide bases occurs due to a gene mutation. We're going to look at gene mutations first. Chromosome mutations come later on when we do cell division and meiosis, or particularly bimeiosis. So, gene mutations are caused by errors in DNA replication. So when the DNA undergoes semi-conservative replication, sometimes, although it's still fairly rare, those bases, or the sequence of bases, may change. Okay. Now, it's important to remember that mutations are spontaneous. Okay? Mutations will happen regardless of any other factor. So, we would never say that, for example, X-rays, UV light and benzene, which we refer to as mutagenic agents, we would never say they cause a mutation. They don't cause, that is all kinds of wrong, because mutations happen anyway. But what they do is increase the rate at which mutations take place. Now, within those mutagenic agents, if we've got a Venn diagram, so we've got mutagenic agents. Inside of that is another group referred to, that's not great, is it, as carcinogens. So these are mutagenic agents that cause disruption to genes, the proteins involved in the cell cycle regulation. Okay, so increasing the rate or regulating the rate of the cell cycle, and in doing that they can lead to tumour development. So carcinogens might lead to tumour development or what we would consider cancer. Now, not all mutations are bad. Okay, natural selection relies on this idea that mutations take place, and some of them are beneficial. If a mutation occurs and there's a change to the sequence of nucleotide bases, and that changes the sequence of amino acids in a polypeptide, and that gives that polypeptide some kind of advantage, what we then have then is a selective advantage, and that may increase the reproductive success. So reproductive success means how often is that individual able to reproduce and pass on their alleles. So sometimes a mutation may increase the reproductive success, such as this example here. So mutations are responsible for variation. So all the variation we see within a population are caused by different alleles, and those alleles are produced by mutations. Any unfavourable mutations that cause an allele which is detrimental to the health of that individual lowers their reproductive success, and those tend to be selected against. Selected against means they stop existing in the population. Favourable mutations will be selected for. Okay, so those individuals with the favourable selection or the favourable mutation are able to reproduce more often, and if they reproduce, if they have a higher reproductive success, that means that more of their alleles are passed on. Now we'll look at natural selection in more detail later, but that is a rough idea. Now, okay, so we'll move on now. Let's see if we can get this to work. Not sure what the copyright implications of using this are, but let's try. So there are three different types of... Is this even going to work? No, it isn't. Never mind. There are three different types of mutation that you need to be familiar with. If we have a look at the notes here, the first one is known as a substitution. So substitution is the replacement of one or more bases by different bases. Okay. The substitution of a single base may result in a few different things. So let's have a look down here. Okay, so I'm actually going to look at this last bullet point first here. So here we have a sequence of nucleotide bases. This is the original sequence, and this is the mutated version. We can see here that this adenine has been substituted by guanine. So here we have the amino acids that are coded for thionine. What is that? I've had a complete mind blank. Anyway, something alanine and glycine. Now, because of what's called the degenerate, that's a D by the way, because of the degenerate nature of the genetic code, i.e. that sometimes more than one triplet codes for the same amino acid. So more than one triplet equals the same amino acid. So after transcription and after translation, this original sequence of nucleotide bases codes for the same amino acid as this sequence of nucleotide bases. So this is a substitution that has no effect. The polypeptide, therefore, remains unchanged. Hydrogen bonds, ionic bonds, disulfide bonds all form in the same position in the tertiary structure, and it has no effect. Let's compare that to this one though. Okay, here we can see again there is a substitution for this time an adenine is substituted with a cysteine, just a single base substitution, but this time that amino acid is now this amino acid. Now the problem with that is that, and this is what you must include in your response. So let's go through this. We can see adenine is substituted by cytosine, whatever that amino acid, I'm so embarrassed, is by that one. So this has changed the sequence of amino acids, or you might say it's changed the primary structure. Now, change to the primary structure will eventually lead to hydrogen bonds or ionic or disulfide bonds forming in different positions. Now this marking point is really important, because if they form in different positions, that changes the tertiary structure. Okay, so the things you need to mention then. What is the change in the nucleotide bases? What is the change to the sequence of amino acids in the polypeptide? You must mention this change to the position or the location that hydrogen ionic and disulfide bonds form, and also the outcome of that is a change to the tertiary structure. Okay, so we get a change to the secondary structure because there's a change to the primary structure, and then a change to the tertiary structure, because these bonds form in different positions. That bit is, I can't emphasize enough how important that is, to say that the hydrogen or ionic or I'll do it with F this time, disulfide bonds in different positions, form in different positions. Okay, so that is particularly problematic. Well, maybe not, but it is problematic for globular proteins. So those globular proteins that have very specific tertiary structures, which enables them to perform their function, would be affected by this change in the sequence of amino acids in the primary structure, changing the position of the hydrogen ionic disulfide bonds in the tertiary structure, and changing their overall structure. So typically we see this for questions relating to enzymes, but also it might be a transport protein, so a channel protein or a carrier protein. It might be for hemoglobin, or it could be for antibodies, that variable region of the antibodies that are complementary to the antigen. I suppose when we talk about antigenic variation, that same principle applies. So if the antigen changes in a virus, for example, it then appears as a new virus and wouldn't be recognised by the immune system. Okay, one example of one of these single nucleotide base substitutions is this here. So this is Sickle-Cell Anemia. Okay, where there is a change to the hemoglobin A. Here is the nucleotide base sequence. Here is the amino acid sequence. If we have a look here, in hemoglobin S, so the mutant adult hemoglobin S, which causes sickle cell, there is a change of the adenine to thymine in the nucleotide base sequence. That changes the amino acid to a valine, and that causes the shape of the hemoglobin to change, which results eventually in the sickle cell red blood cells. And they've got a lower capacity for carrying oxygen, and they also increase the likelihood of clots forming and aneurysms. Okay, so the next type of what am I talking about again? Mutation is a deletion. So deletion involves a removal of one or more bases. Now this is problematic. I don't have it on here because it causes what's called a frame shift. Now frame shift means, well, if we have a look at this diagram down here, let's try and expand on this, make it a bit bigger. Okay, so here is the normal. Here's the normal DNA. So this is before the mutation, and we can see a deletion has taken place here. So a deletion means one of the bases has been removed, and what we notice is that after that point of deletion, we get this frame shift. So all of the subsequent amino acids after all of the subsequent triplets after the deletion will be changed. And that means all of the subsequent amino acids that would be coded for after that deletion have also been changed. Now this obviously has a much greater effect on the protein and is almost always, or results in almost always, a protein that is non-functional. It may even introduce a stop codon earlier than is necessary, so that the protein may be shorter. Again, that would result in it being non-functional. An addition, you've probably worked out what an addition is where one or more bases is added in. So here we can see ATT, CC, GT. ATCC is the same, but then we've got this addition of one or more bases, so a thymine and a cytosine are added in here. And again, this would cause a frame shift, which means all of the base triplets from that point of the addition would be altered. Okay, so that means the sequence of amino acids is altered from the point of the addition, and the protein, again, is almost always non-functional. A couple of other types. So duplication is where, what's going on here? One or more of the bases gets repeated. So we can see that here. We can see that cytosine guanine is repeated. Okay, this also causes a frame shift, and again, all of the triplets from that point of addition are changed. An inversion is where the sequence of bases gets reversed. So flip backwards. That's going to change the amino acids that are coded for at that point of inversion, which I guess, depending on the length of that inversion, will dictate how functional that protein is. And lastly is a translocation. So this is where one of the sequences is actually moved from one location in the DNA, so one location on the chromosome, to another part of the genome altogether. So perhaps to another part of the chromosome, or perhaps even to another chromosome altogether. Okay, let's go back to our PowerPoint here. So, genetic diversity depends on those mutations. So the variety of alleles in a species is referred to as the genetic diversity. Okay, a population is all of the individuals of the same species in a habitat or in an area, in a given area. And therefore, species or population here is fine for this definition of genetic diversity. Okay, so organisms of the same species have the same genes, but we know that there are different versions of those genes, alternate versions of the genes that we call alleles. So, whilst those organisms of the same species have the same genes, they won't have the same alleles unless they are a identical twin. With increased number of alleles in a particular population or species, that increases the genetic diversity. So all of those alleles in that population, in that species, combined together is what we refer to as the gene pool. So gene pool is all of the alleles of a species. Okay, and that is important. So this genetic diversity is important because a more genetically diverse species or population is more likely to survive a change, because it's likely that within that population there are some individuals which have alleles which favour perhaps environmental change, and whilst others don't have those alleles, the ones that do have a selective advantage, they can survive, they can reproduce more often, pass on their alleles. So a wider variety of alleles means it's more likely that some individuals will be adapted to survive the change. Typical changes might be environmental, new diseases, new predators, or new competitors. And that's where we'll finish there. I think I've rambled on for long enough. I'll come back in a minute and we'll do another video on cell division. Okay, thank you.